Hello my dear students, a very good afternoon to all. This is Vasavi Ma'am, your biology educator for Foundation and CBSE. Yes, students, in the last class, just I'll quickly revise you what we have discussed in the last class, children. Okay, in the previous session we have just discussed about the composition of the plasma membrane, as well as we have also studied in detail about the transport mechanisms like active transport and passive transport. Okay, so children, just once I will revise you in a short time. Okay, so the plasma membrane is actually made up of a phospholipid bilayer, which is going to contain, if we look into the single lipid structure, it is made up of the hydrophilic head and hydrophobic tail. Okay, so hydrophilic head, in the sense it is going to remain in close association with the solvent molecules, and the hydrophobic tails are not in contact with the solvent molecules. Okay, and next we have also discussed that in between the plasma membrane there are some scattered proteins. Okay, so the proteins are actually meant for carrying the ions or molecules from the outside of the cell environment to the inner side of the cell environment. Okay, it is just going to span the plasma membrane and it is involved in transporting the ions or molecules. And next, along with these integral proteins, along with these intrinsic proteins, there are also some proteins which are present outside of the, which are going to remain in association with the hydrophilic heads. Okay, those are called as extrinsic proteins. Okay, and here we have the carriers. Okay, we have the channel proteins, okay, which are meant to transport the ions like sodium ions or potassium ions. Okay, now if we look into the transport mechanisms, that is active or passive. So when it comes to the passive mode of transport mechanism in the plasma membrane, so passive transport mechanism is of two types. It may be either simple diffusion or else it may be either facilitated diffusion. Right, children? So in the simple diffusion, the molecules are going to move from the high concentration gradient to the low concentration gradient. So here you need to remember that here we are not going to supply any energy. Okay, here we are not going to give the energy in the form of ATP. Okay, so the energy is going to be absent. Okay, in the absence of energy, the molecules are going to move randomly from the high concentration gradient to the low concentration gradient. Okay, that we have discussed in the passive mode of transport mechanism, which is called as simple diffusion. Okay. So in the facilitated diffusion, we have discussed that some proteins, okay, some carrier channels are required. Okay, some channels are required, some way of transport is required to transport the molecules or ions from the outer cell surface to the inner cell surface. Okay, in the facilitated diffusion, we have studied channel proteins and carrier proteins, okay, which are involved in transport mechanism. Yes, when it comes to the active transport mechanism, in the active transport, we need to supply the energy in the form of ATP. Okay, in the active transport mechanism, what we have to supply, children? Their ATP, their energy is required to transport the ions or molecules against the concentration gradient. Okay, that for that we have discussed some channels are sodium and potassium pumps. Okay, so their special pumps are required to transport the ions or molecules. Okay, to transport the charged ions or molecules, so the potassium pumps and sodium pumps are required. Right, children? So that potassium, so outside of the cell surface, okay, outside the plasma membrane, the concentration of sodium is more. Okay, inside the cytoplasm, the concentration of potassium is more. Clear? So the potassium is less in the outside the cell surface. Okay, outside the plasma membrane, the concentration of potassium is less. Okay, so it is always easy to transport the molecules or ions from the high concentration to the low concentration. But here in the active transport, we need to transport from the lower concentration to the higher concentration gradient by the involvement of the energy in the form of ATP. That is all about the active transport mechanism and passive transport mechanism in the plasma membrane. Okay, so now we are going to study few more transport mechanisms. Those are called as osmosis and the next bulk transport mechanism. Yes. So before discussing those topics in brief, so now we will just try to solve some questions regarding the plasma membrane structure. Yes. Yes children. So once I will show you this animation video, by that it gets clear for all of you how the ions or molecules are going to be transported. Yes. See children, if you look here, this is what it is. It is a channel protein, okay. It is transporting the ions, okay. It is transporting the water molecules, solvent molecules from the outer cell surface to the inner cell surface. Clear now? Clear with the concept? Okay. Here the solvent concentration is more, so it is transporting the molecules or ions from the higher concentration to lower concentration gradient by the involvement of what? By the involvement of channel protein. Yes. Yes, here comes the case study question, okay. So if you are clear with the concept, the plasma membrane structure and its composition and its mode of mechanism, okay, and its mode of transport mechanism, then it becomes easy to attempt these questions children. Yes. So here the question is that identify A, B, C and D in the given figure X, okay. We have to identify the labeling parts A, okay, and then next B, C and D in the given figure X. So now it is a question to all of you. So please try to answer what is the given what is the figure X here, okay. What is this figure X displayed here children? So the X is actually a plasma membrane, okay. So the X is a plasma membrane and here this Y is the phospholipid bilayer. This total figure X is actually called as plasma membrane, or else it is also called as the semi-permeable membrane or selectively permeable membrane, okay. So now here we have to identify the labeling parts A, B, C and D. So now first let us look into the A, okay. So A is what is the A represents here? A is hydrophilic head, okay. So the option A is, sorry, the labeling part A is hydrophilic head, hydrophilic head, okay. And next B represents hydrophobic tail, hydrophobic tail, okay. And then next D represents what children? D represents extrinsic protein, extrinsic protein, okay. Yes. And then next here C represents intrinsic protein, intrinsic protein, okay. Now clear with all the labeling parts. Now keeping all these concepts in mind, you need to solve these case study questions. Here are the options, so you have to choose the correct option which is relevant to the given figure X here, okay. Yes. So here option A, okay, in the option A, so A, he is telling that hydrophobic tail, okay. Here the option A is given as hydrophobic tail, but actually what is option A children? Option A is hydrophilic head, so it is not hydrophobic tail, so the given option A is wrong, okay. And next B is hydrophilic head, okay. What B represents here? Is it hydrophilic head? No, it is hydrophobic tail, okay. It means it is water hating, hydrophilic head in the sense water loving. So again here option B is also a wrong statement, yes. And then next what does the option C represents, okay. See here the given option C here is intrinsic protein, but actually yes, C is intrinsic protein, yes. So the C is correct, but we need to choose all the options which are correct, okay. So here C is correct, yes. And next D is extrinsic protein, yes, this is also correct. So but here two options are correct and two options are incorrect, okay. But we have to give the correct options in an arranged manner, okay. Along with this, okay, here whatever you can see the A, B, C and D, you need to give the correct option regarding with this particular figure X, okay. Yes. Now next coming towards the option B, here A, he is given the option as hydrophilic head, yes, A is hydrophilic head, it is correct. And next B, hydrophobic tail. Yes, B is also correct. And next C is extrinsic protein. Actually C is what, children? C is intrinsic protein. Okay, so it is a wrong option. Okay, and next coming towards the D. D is intrinsic protein. Okay, actually in the plasma membrane structure, the D represents what, children? The D represents the extrinsic protein but not intrinsic protein. So this option is also wrong. Here also we have classified and we have cleared the 2 options are correct and 2 options are incorrect. So this option B is also going to be a wrong option. Yes. And next coming towards the option C. So here the given option A as hydrophilic head. Yes, if we see the option A, it is hydrophilic head. Yes, it is a correct option. And next B is hydrophobic tail. Yes, absolutely. B is called as hydrophobic tail. So this is also a correct option. And C is intrinsic protein. Yes, if you look into this structure, see here. Okay, this scattered protein, this is called as the intrinsic protein. So here this is also a correct option. And next D is extrinsic protein. And next, yes, in the options also the given option D is extrinsic protein, that is also a correct option. Okay, have you observed in the option C all the options are correct. Okay, A is hydrophilic head, B is hydrophobic tail, and next C is intrinsic protein, and then next D is extrinsic protein. So among the option A and B, we got the correct option C. C will be the correct answer, children. Okay, C is the correct option. So now let us look into the option D. Yes, hydrophobic tail. A is not a hydrophobic tail, it is actually a hydrophilic head. And then next B is hydrophilic head. Yes, this is also a wrong option because it is an hydrophobic tail. And C is extrinsic protein, this is wrong. And next D is intrinsic protein, this is also wrong. So these case study questions, okay. So in this manner, okay, in this way you are going to get the case study questions based on the concepts, children. Okay, you need to understand the given concepts clearly. Okay, its composition you need to understand. In the plasma membrane, it's each and every structure you need to study. Okay, when I explained the structure, there we have discussed about the single lipid structure. Okay, it is actually made up of 2 components, we said. Okay, one will be the hydrophilic head and the tail part will be the hydrophobic tail. Okay, and next it is made up of the few proteins, okay, which are inside, those are called as intrinsic protein, and some are remained in association, some are going to be attached with the hydrophilic heads, those are called as extrinsic protein. Okay, so in this way you need to learn everything. Okay, you need to learn in detail about its structure as well as its composition. Okay, this is the first case study question from your NCERT. Yes, and next now we look into one more case study question, children. Yes. So the second question is, the X is mainly composed of. The X is mainly composed of. What is X again? It is a plasma membrane, right? So here the question is based on the composition. Okay, just now we have discussed. So you need to answer this particular question. Yes, the given options are protein, lipid, and the next cellulose layer. Okay, it is made up of a phospholipid bilayer, right? So proteins and lipids, it is made up of few intrinsic proteins and the next lipid layers. Okay, so the option in option A, proteins and lipids are correct, but here again one more statement is there, that is called as cellulose layer. Okay, what is cellulose layer, children? What is cellulose layer? So cellulose layer, cellulose is a cell wall which is present in the plants. Okay, cellulose is a cell wall which is present in the plants. Here the cellulose layer in the cell only we have the cell membrane, outside the cell membrane only we have the cell wall, that is called cellulose, right? So here it is only, we are discussing only about the cell membrane but not the cell wall. So option A is wrong. Yes. Next, option B, lipid bilayer and proteins. Do you think whether it is going to form a correct answer? Yes, let's stay tuned. Okay. Next we are going to discuss about the option C, protein layer between 2 lipid layers. Okay, we have the arrangement of phospholipids bilayer. Bilayer means in 2 layers the lipid layers are going to be arranged. This is one layer and then next this is one layer. In between these, can you see the continuous arrangement of proteins? Continuously proteins are present? No, okay. So continuous arrangement of proteins are not there, but proteins are present in between them. In between the lipid bilayer we have the proteins, intrinsic proteins are present, and outside that we have the extrinsic proteins, but continuous arrangement of proteins is not there. So in between two layers there is no protein layer. So this is also a wrong option. Yes, next D is lipid layer between two protein layers. Okay. So this is a very simple option, you need to analyze whether it is a correct option or wrong option. What is this option? Lipid layer between two protein layers. Okay. Do we have the two protein layers here, children? Can you see the two lipid bilayers here? No. Okay. So actually the the plasma membrane is itself made up of phospholipid bilayer, but not in between two protein layers. Okay. We have two lipid layers, but not two protein layers. Proteins are just present, those are just going to span the plasma membrane, which are just involved in transporting some passages. Okay, those are just meant for transporting some things from outside to the inner side. So now based on these all options, which will be the correct option, children? The option, yes, option B is the correct option. Okay, protein, yes, lipid bilayer and proteins, yes. Plasma membrane X is mainly composed of lipid bilayer and the next proteins. Clear with the concept? Clear with this case study question? Yes. Yes, one more question. Okay, which component of X completely spans the lipid bilayer and forms the passage for substance? Okay. So directly, okay, here you have the direct answer, option C. Okay, the labeling part C. What is C? Intrinsic protein. Okay, it is meant for passage of the ions or substances from the extracellular fluid to the intracellular fluid. So now if you look into the options, yes, intrinsic protein is the correct answer. Okay, whereas option A, extrinsic protein, wrong, and next option B, polysaccharide. Okay, it is also wrong. Those are present, but those sugar molecules are present on the lipids as well as proteins. Okay. So this is also a relevant option when we discuss about this particular question. Yes, next C is intrinsic option. Yes, this is the correct one. And next D is phospholipid. Okay, phospholipid is not going to span. Okay, it is continuously arranged. But here the protein, intrinsic protein is present, which will span, which will cover some portion in the plasma membrane, and it will help in transporting the substances. Yes, clear with the concept? Yes. Clear with all the questions regarding the plasma membrane structure as well as its things. Okay. And next we are going to discuss about the one more method of transport mechanism in the plasma membrane. Okay, diffusion is completed. We have discussed about the diffusion, active transport, and the next passive transport. In the passive transport again we have discussed clearly about the simple diffusion and the next facilitated diffusion. Now here there is a one more type of transport mechanism, that is called as osmosis. Okay. So children, please stay tuned and please try to understand the concept clearly. Based on these also you need to solve one case study question. Okay. It is very easy topic, but it will be somewhat critical to understand. Okay. So I will make you the way to learn easy, children. So please focus clearly. So when we discussed about the diffusion, in the diffusion what we have discussed, children, just this will be, suppose it is a plasma membrane. Yes, I'll make it clearly for you. Yes, when it comes to diffusion, okay, here suppose this will be the plasma membrane, okay, and these are the hydrophilic heads and these are hydrophobic tails. In diffusion we have said that molecules are going to move from high concentration gradient to low concentration gradient without involvement of energy and without involvement of the plasma membrane. So directly randomly they are going to diffuse from inner to outer side of the cell. For this best example is exchange of gases in humans. Okay. Respiration will be the best example. So now when we discuss about the osmosis, when we discuss about the mode of transport mechanism, that is osmosis, here molecules are going to move from higher to lower concentration gradient. See here, high concentrated molecules are present. Okay, molecules are in high concentration gradient, and here very few molecules we have. So in the osmosis, molecules move from high concentration gradient. Okay, which molecules? Only solvent molecules. Okay, only the passage of solvent molecules from higher concentration gradient. Okay, only water molecules are involved, children. Okay, only solvent. Okay, so the movement of solvent from the higher concentration gradient to the lower concentration gradient by the involvement of semi permeable membrane or selectively permeable membrane. Okay, by the involvement of plasma membrane, by the involvement of this cell membrane, the molecules, the solvent molecules are going to move from high concentration gradient to the low concentration gradient. So if you see, if you observe in the diffusion, okay, in the diffusion any type of molecules can be moved. Okay, any type of molecules can be transported. Okay, that may be either solids, liquids or gases, but here when it comes to the osmosis, we can transport only the solvent molecules from higher to lower concentration gradient, and here we need the plasma membrane, we need the selectively permeable membrane to transport that. Okay, semi permeable membrane is required to transport the solvent. Okay, yes, and then next, if we look here, osmosis is also a passive movement of water. Okay, osmosis is also a passive movement. Here also we are not going to supply any energy. Okay, we are not going to supply the energy in the form of ATP. So it is also a passive transport mechanism. Yes, and here any other solvent molecules, they are going to move from a region of high concentration to a region of the low concentration gradient. Okay, by the involvement of semi permeable membrane. Okay, clear with the concept, clear with the definition osmosis? Yes, and next, osmosis can be of two types. It may be either exosmosis or else it may be either endosmosis. Clear with this concept up to here, whatever I have explained you? Yes, osmosis is of two types, exosmosis and endosmosis. Okay, it can be observed in these three cases. Okay, so to understand the two types of osmosis process, okay, that is exosmosis and endosmosis, you need to study the three types of cases. Okay, you need to study the few short experiment to understand you in a better way. I will explain you, children. Okay, so you need to understand these concepts. Okay, you need to understand the types of solution to study the exosmosis and endosmosis. Yes, those are isotonic, hypotonic and hypertonic solutions. So now clearly, so please everyone slowly listen the concept and please note down whatever I am explaining you, children. Yes. Yes. So I request everyone to note down this concept, by that it becomes easy to all of you. And once again I am informing you all, children, this concept is easy but it is very confused to be understand. Okay, so please listen whatever I am explaining you. Yes. So this is a beaker. Okay, this is a beaker A. Yes, let me draw it once again here clearly. Yes. So take one beaker, okay, and then next label it as beaker A. Beaker A. Okay. So in the beaker A, you add some, yes, you need to add some solvent. Okay. What is this all, children? Whatever I am drawing it in a blue color manner, this is all the solvent molecules. So before understanding this in clearly, okay, so to make it in easy way, you need to study some terms. Okay, those are solution. Okay, you need to understand what is solution. Okay, and then next, you need to understand the terms solutes and next solvents. Clear? So when you understand these three terms, it becomes easy to understand the process of osmosis. So first look here, solution. What is solution? Solution is made up of two components, those are solutes and solvents. Yes, now we will discuss what are solutes. see, solutes may be any type of molecules, okay. They may be either sugar or salt, okay. So, the solutes are the substances which are going to dissolve in the solvent, okay. Now, this is the solvent. What will be the solvent, children? Solvent will always be water, okay. Solvent is what here? What is the solvent here? Solvent is water, okay. So, now solutes are the substances which are going to dissolve in the solvent. Now, what is solvent? Solvent is the substance in which the solutes are going to dissolve, okay. So, this is actually a solution, okay. By getting the clear idea about these terms, so you can understand the exosmosis as well as endosmosis, okay. So, osmosis is the movement of the molecules, okay, the movement of solvent molecules from higher to lower concentration gradient by the involvement of semi-permeable membrane, okay. So, for this we have going to study 3 types of solutions, okay. To understand the osmosis, we are going to discuss 3 types of solutions, okay. So, to understand, so before learning about that solutions, before learning about those 3 cases, first we are going to discuss about these terms. What are the solution? Is made up of solutes and solvents, okay. Solutes are nothing but the components or else the substances which we are going to dissolve, okay, in the solvent, okay. The solutes may be either sugar or salt. So, the solvent will be the water. So, both together will form the solution. So, now I will explain you a general experiment, okay. So, now we will study about the 3 types of solutions. Yes. So, children, please note down these points. Yes, this is a beaker A, okay. So, in the beaker A, you need to add water, you need to add the solvent plus solute. So, children, now see here, whatever the blue color lines I am drawing, those are called as solvent, it is solvent. I am adding water here. It is a general experiment which you can perform in your home also, okay. So, it becomes easy to understand the concept. So, in this beaker A, I have placed the water, okay. I have poured some water, okay, that is nothing but solvent. Let me label it here clearly, okay. This blue color one is solvent, okay. And along with this solvent, I am going to add some solutes. What may be those? That may be either sugar or salt, okay. Let me take this red color, okay. So, these things, okay, whatever I am drawing here now, red color one, these are all the solutes, okay. It is the solutes, okay. The solutes may be either sugar, that may be either sugar or salt. So, now in this solution, okay, in this solution, that is nothing but we have already filled the beaker with some water along with some solutes. So, now here we are going to place one resin, okay. So, can you see it is going to become confused, I think. I will draw it once again, children. For the better understanding, let me make it clear once again. Yes. Yes, this will be the beaker A, okay, in which we are going to dissolve the solvents and solutes, okay. This is a solution actually, which is composed of both the solutes and solvents. Yes, this blue color structure, whatever I am drawing now, these are solvent molecules. It is a solvent, okay. Along with the solvent, I am going to add the solutes, that may be either sugar or salt. Yes, I am adding the solutes, okay. So, now observe clearly here in this beaker, the solutes are more, okay. The solute percentage, the solute concentration is more when compared to the solvent. Yes, this will be the solutes, that may be either sugar or salt. So, the percentage of this solutes is high, okay. The percentage of solutes is high And next here in this beaker we are going to place one resin. Now we are studying all these cases. Now we are studying the types of solution. You will understand later when I'll be completing this concept. Okay, then you will understand which type of solution actually it is. So now we are studying the osmosis, right? So in the osmosis we have said that by the involvement of semi-permeable membrane only the molecules are going to cross. Okay, the molecules are going to pass from higher to lower concentration gradient. So here it is nothing but we are studying all these types of solutions with respect to the cell, children. Okay, we are studying these types of solutions with respect to cell. So now here I am going to place a resin. Okay, here I am going to place a resin. So now please clearly understand, please try to understand here the solutes percentage is more, okay, with respect to the cell. Inside the cell, yes, inside the cell the solvent is less. Okay, inside the cell the solvent is actually more, children. Okay, inside the cell we have the more solvent. Okay, but outside here we have solutes more and inside we have the solvent more, but in the beaker the solvent percentage is less. Clear with the concept? We have took one beaker and we have filled it with, we have created one solution, we have prepared one solution. Okay, we have added the solute molecules, that may be either sugar or salt. Along with that we have also again added the solvent, that is water. So again we have placed one resin. Okay, so here in this complete outer, so to the cell environment the solute percentage is more. Okay, and inside to the cell the solvent is more, and in this solution the solvent is less. Clear with the concept? So now here you need to understand, in the osmosis always solvent molecules will move from higher to lower by crossing the semi-permeable membrane. So now I am saying that here solvent is more inside the cell. It means what happens? The molecules, the solvent molecules will move from higher to lower concentration gradient. Clear with the concept? Here the solvent is less outside the cell. Okay, inside the cell solvent is more and solute percentage is more outside the cell. So we have to transport the solvent molecules. So inside the cell solvent is more. Okay, so always the solvent will move from higher to lower concentration gradient. From here to here it moves. Okay. So at this particular case what happens? Okay, it is a case 1. Okay, now you need to give me the answers and you need to give me the conclusion based on these types of solution, children. Okay, so here what happened? All the solvent molecules which are present inside the cell, they are going to be pushed outside. Okay, they are going to be transported outside. Okay, so at this time the cell is going to shrink, the cell is going to lose the water. Actually in the starting stage when we placed the resin, it was in this structure. Later by losing the water it became shrinked. Okay, so now all these study, whatever we have studied, this every concept, okay, the process whatever it is happening in this type of solution, it is called as hypertonic solution. What, children? It is called as hypertonic solution. Hypertonic solution. Clear with the concept? In the hypertonic solution the concentration of the solutes is more. So when compared to the cellular environment, inside the cell the solvent molecules are high. So outside the cell environment the concentration of solvent is less. So the solvent will move from the higher concentration to the lower concentration gradient by crossing the semi-permeable membrane. So during this process all the solvent molecules will move outwards. Okay, all the solvent molecules will move outwards by removing the water, by removing the solvent. So this process is called as exosmosis. Okay, so this process is called as exosmosis. Okay, based on this again you need to solve one case study question, children. So please try to remember exosmosis in the sense the outward movement, okay, the diffusion or else the outward movement of the solvent molecules from higher to lower concentration gradient. Okay, so here all the solvent molecules will move outside, and next So due to that reason the cell becomes shrink, okay, the cell becomes shrinkage, okay, this is called as shrinkage of the plasma membrane. You need to study, okay, you will study in the next slide. Understood about the hypertonic solution? In the hypertonic solution, the percentage of the solutes, the concentration of solutes is more. Inside the cell we have the solvent more, so we need to transport, okay, always the solvent will move from higher to lower concentration gradient. So that transport mechanism will lead to the exosmosis, which leads to the shrinkage of the plant cell, okay, shrinkage of the resin. Yes, now next we are going to study about the hypotonic solution. Next, hypotonic solution. Yes. So in the previous, in the first case we have took a solution, okay. There we have in the solution we have the solutes plus solvent. So now we are going to take a simple solution, okay. Now we are going to, we are not going to consider it as a solution, but instead we are going to take one beaker and we are going to fill it with the solvent. I am going to fill it with completely with the water, okay. Okay, I am going to fill this beaker completely with water without adding any solutes. So here again I am going to place a resin. Students, this is a simple activity which you can perform in your home itself, and you can perform this activity also, but a little bit confusion, you need to understand the terms properly, then only the concept becomes easy to you. Yes, here again I placed one resin. So here there is no solutes, solutes are absent, okay, just I added only solvent, okay, here only solvent percentage is more. So when compared to the cell environment, inside the cell the solvent is less. Now you need to answer, solvent is less here inside the cell. Outside to the cell we have more solvent, concentration of the solvent is more. So now from where to where the solvent molecules will move? Absolutely from here to here, okay. The solvent will move from higher to lower concentration gradient, okay. So outside the cell environment the concentration of solvent is more, hence it moves from here to here, okay. Hence the solvent molecules are going to diffuse from outer cell surface to the inner cell surface, okay. Here the process of mechanism you can observe is endosmosis, okay. In the endosmosis the cell will gain the water. In the endosmosis what happens, children? Please try to pay your attention and try to understand the concept. In the endosmosis the cell is going to gain the water, and in the exosmosis the cell is going to lose the water. That exosmosis we have observed in the hypertonic solution, and endosmosis, this complete process we can observe in case 2, that is hypotonic solution. Hypotonic solution. Clear, children? So at this particular case what happens to this cell? It is going to take all the solvent molecules, okay. Here inward movement of water is, inward movement of solvent molecules is going to occur. At that time, during the starting initial stage the resin was in this size, okay. Just imagine it was in a medium sized, okay. So after placing it in the hypotonic solution, the cell gets swelled, cell swells. We can give the term cell swells in the hypotonic solution, cell swells in the hypotonic solution, okay. How it is going to swell? Actually it is going to swell by endosmosis, it is going to swell by endosmosis. It is nothing but inward movement of the solvent molecules. Cell shrinks in the hypertonic solution, that is just due to the exosmosis, just the outward movement of the water molecules or solvent molecules, okay. So in this complete case study, case 1 and 2, we have observed how the cell is going to gain the solvent molecules and how the cell is going to lose the solvent molecules, okay. We have studied all these two types of solutions, okay, with respect to the cell. And next here is one more case, okay. Here you need to study case 3, okay. Yes, in the case 3 what happens? Again you need to place a resin along with the solutes and solvent. Yes, this is a beaker in which I am adding the solvent along with the solutes. The red color portion whatever I am drawing, these are the solutes, and here again I am placing the resin. Okay, clear? So here what happens, the solute concentration and solvent concentration which is present inside the cell, okay, the solute and solvent concentration with respect to the intercellular, okay, inside the cell, both the concentrations are same when compared to the extracellular fluid, that is nothing but this outer cell environment. Okay, here whatever the solutes plus solvents, okay, whatever these are present outside the beaker, those are equal to the solutes and solvents which are present in the cell, both are equal. So there will be the equal movement, okay, there will be the net movement of solutes and solvents from the cell membrane. Okay, so here whatever the solutes and solvent is going to transport outside the cell, okay, outside to the cell is equal to the solvent molecules which will enter the cell. So this particular case 3, okay, this particular case whatever we are studying, it is called as isotonic solution. Isotonic solution. Yes, clear with the all the concepts, children? Yes, in the isotonic solution the cell remains same, okay, because the net transport, okay, the movement of the solute, the movement of the solvent from the outside cell to the inner side of the cell will be equal. Okay. So here in the hypertonic solution what happens, here what we have in the hypertonic solution, what we have studied, the concentration of the solutes was more, so inside the cell the solvent was less, so we need to transport from higher to lower concentration. So in that particular case the cell shrinks. Okay, cell always shrinks in the hypertonic solution and cell always shrinks in the hypotonic solution due to the endosmosis, and in the hypertonic solution we have observed the type of osmosis that is called as exosmosis. Clear with these concepts, children? Yes. Is it easy to remember or not? Okay, it becomes easy when you are going to write the notes and when you are going to remember it repeatedly. Yes, these are 3 cases which we studied with respect to this cell. Okay, with respect to the cell we have studied 3 types of solutions. Now here comes the case study question. Yes, before learning about, before solving the important case study questions, once again let's have a look on this, children. By that it becomes easy to you. Yes, osmosis. Slowly clearly observe here. So here see, when compared to the cell, the outer side, okay, the solvent present outside the cell is more. So the solvent is moving from outer to the inner cell environment. So that is the reason the cell is going to swell here. Okay, see, cell swells here due to the entry, okay, due to the entry of solvent molecules, that is endosmosis. Here cell shrinks. Okay, why the cell shrinks? Due to the high concentration of the solvent molecules, everything is going to be pushed outside. Okay, outward movement of the solvent is going to occur, hence it is going to shrink. In the isotonic solution, there will be the equal movement, okay, equal movement of the solvent molecules. Okay, clear with the concept? It becomes easy to you by seeing this particular slide. Yes. Yes, plasmolysis. So this is also a short topic, whatever we have discussed it is the same. Okay, plasmolysis is nothing but shrinkage of the plasma membrane, okay, shrinkage of plasma membrane, that is due to, yes, that may be either due to the hypertonic solution. Okay, here the plasma membrane shrinks due to the exit of the water, due to the exit of the solvent molecules the cell shrinks in the hypertonic solution, and here the cell swells. Okay, that particular case we are going to call that cell as become turgid. Okay, turgid in the sense due to the entry of the solvent molecules, due to endosmosis the cell becomes turgid, and here the cell we called it as flaccid. Okay, where there is no shrinkage is observed and no turgid stage is going to be observed. Okay. So, this is called as the flaccid. Here we cannot observe the whether the cell is plasmolized, we cannot call that cell whether it is plasmolized or whether it is turgid. Okay. Here because there will be a equal movement, okay, there will be equal transport movement of the solvent molecules, and here we are going to call that particular cell as being plasmolized. Okay. We are calling the cell has been plasmolized. Why we are calling the cell as plasmolized? Because due to the exosmosis the cell, okay, the plasma membrane is going to get shrink, that is the reason we are calling it as the plasmolized. Okay. This is the plasmolysis concept. Okay. Same whatever we have discussed that only, but with respect to cell we are going to give these particular names, okay, depending upon the solutions only. Yes. Yes, here comes the case study question to all of you. Yes. Select the incorrect statement. Okay, select the incorrect statement. Yes, solution A, B, C, or else we can also write it as beaker A, B and C. A, B and then next C. Yes. So, here in beaker A, what you are going to observe? Select the incorrect statement. So, first let us have a look on these options whatever given here. See, A, beaker A, okay, option A the given here is, in the solution A, the medium surrounding the cell has high water concentration than the cell. Okay. So, here in the solution A, the given option is the medium surrounding the cell has high water concentration than the cell, okay, than the water concentration than the cell. When the water is higher surrounding the cell, what happens, children? All the solvent molecules are going to enter inside the cell. Okay. At this particular case, what happens to that particular cell? It is going to swell, right. So, this particular type of solution, what we are calling this type of case, it is called as hypotonic solution. Remember, hypotonic solution. Yes. Next, solution B. Here, due to exosmosis, due to the loss of water, due to the loss of solvent molecules, it is going to become shrink. Okay. In which case we have observed the shrinkage of the cell? In the hypertonic solution. And this is the option C, which we call it as isotonic solution. Yes. Now, you have to tell me whether the option A is correct or not. In the solution A, the medium surrounding the cell has a higher water concentration than the cell. Yes, this option is correct, but here we need to identify the incorrect statement. Yes. So, this according to this question, this is a wrong option. And next, option B, in solution C, the external medium has exactly the same water concentration as the cell. Yes, absolutely right, because it is isotonic solution, where there will be the equal concentration gradient inside the cell and outside the cell. Yes. Next, option C, in solution B, the cell will lose the water due to endosmosis. Okay. What is endosmosis, children? What is endosmosis? Endosmosis in the sense loss of, yes, gaining the water. Okay. So, but here in solution B, cell will lose the water. Cell will lose the water due to the process of exosmosis. Okay. Cell will lose the water due to exosmosis. Okay, but not endosmosis. So, here the correct option is option C. In solution B, the cell will lose the water due to endosmosis. Okay, that is a wrong option. Cell is losing in the sense cell is just ejecting the solvent molecules from the inner side. So, that process is called as exosmosis, but not endosmosis. So, option C will be the right option. Option D. Yes. In option D, in solution B, the surrounding medium has a low concentration of the water than the cell, making the outside solution concentrated. Yes, this is also an irrelevant option when compared to this particular question. Yes, clear with this particular case study question? Yes. Next one. Next case study question is, when a cell is placed in a solution A, there is a gain of water. Gain of water in the sense hypotonic solution. Okay, clear with the concept? Always the cell is gaining the water, in the sense there the endosmosis process is going to occur. In that particular case the cell is going to swell. So, we can observe this process in the type of solution called as hypotonic solution. No doubt it is an direct answer. Yes, isotonic wrong, hypotonic is also a wrong, and concentrated solution, this is also a wrong statement. Next question, in solution B, the water moves out through the process of exhaust, through the process of, in the solution B. Okay, here it is losing the water, losing the solvent molecules means absolutely the answer will be the exosmosis. Yes, clear with the concept? Yes children. Yes, next here comes one more type of transport mechanism, clear with the osmosis. Okay, clear with the osmosis definition as well as its types, that is exosmosis and endosmosis, and we have studied the osmosis with we have studied in 3 cases. Okay, in 3 particular solutions. By taking the 3 solutions we have understood the osmosis concept with respect to the cell. Okay, so now here there is a one more one more mode of transport mechanism, that is called as bulk transport mechanism along the bulk transport mechanism related to the plasma membrane. Okay, bulk transport mechanism across the plasma membrane. Okay, so in the bulk transport mechanism, what we are going to transport? We are going to transport the large macro molecules. Okay, large macro molecules and some large food molecules, large food particles, and the next large macromolecules we are going to transport. Okay, that mode of transportation is called as bulk transport. Macromolecules and some food, large food particles. I am writing it as a P, food particles and macromolecules. Okay, these two we are going to transport. Okay, so this process of transportation is called as the bulk transport. It is of 2 types, exocytosis and endocytosis. Exocytosis and endocytosis. Yes, so first you try to understand what is endocytosis. Look here, look into this picture clearly children, so that it becomes easy to understand. Yes, endocytosis. Endocytosis is of 2 types. Okay, one is phagocytosis and one more pinocytosis. Okay, these all we are studying under which transport mechanism? Under the bulk transport mechanism. So remember, bulk transport mechanism is also active mode of, okay, active way to transport the large food molecules and food particles along with some large macromolecules. Okay, here also you need to transport the energy. Okay, this is also an active mode of transportation. Yes, endocytosis is of 2 types, one is phagocytosis and next one is pinocytosis. So now you look here, the phagocytosis is also called as cell eating process, cell eating transport. Okay, we can also simply call it as cell eating process. Okay, so here what happens in the sense, so this we are studying in amoeba. Okay, this is an example how the phagocytosis is actually going to occur in amoeba. Yes, so here are some food particles, large macromolecules are large food particles which are present here. Okay, so these inside these amoeba we have lysosomes. What are lysosomes? About lysosomes also we are going to study it in detail. So, but actually the lysosome process and its function is, it is actually single membrane bound cell organelle which is involved in digesting the cellular waste materials. Okay. Yes. So, these are the lysosomes and it is a nucleus and this everything is the cytoplasm. Okay. So, here is the food particle. In the next process, this will be the plasma membrane of this amoeba. Okay. So, the plasma membrane will invaginate the food particle. Okay. It will cover the entire food particle. Can you see here? It is going to invaginate like this. It is going to cover the entire food particles like this. Okay. So, in between here we have some small cleft or some small gap like structure. So, in the next stage this is going to be pinched off. Whatever the food particle it is going to take, whatever it is going to invaginate, it is going to cover, and there one vesicle is going to form. Okay, one vesicle will form and it will get separated from the plasma membrane, and that vesicle is going to transport into this cytoplasm. Okay, see here fusy formation of food vacuole within the cell. Okay, after fusing, next what happens? Fusion of lysosomes with the food vacuole. Okay, so inside the cell what is going to be present? Lysosomes are going to present. Okay, so first the amoeba is going to surround the food particles, and next it is going to form one vesicle. Inside the vesicle the food particle is going to be present, and then next those food particles are going to fuse with the lysosomes. So next later all these food particles, whatever it is going to be formed in the vesicle, it is after fusing it is going to digest the food particles. See here, digestion of the food particles. So this is a process, it is called as cell eating process, or else it is also called as the phagocytosis, which is simply called as cell eating process, where it is going to take, where it is going to feed, and where it is going to capture the large food particles. Okay, in the first case here it is a food particle, and inside this we have the lysosomes, and in the next stage it is the plasma membrane is going to invaginate the food particles. Next it is going to form the vesicle, and then next it is going to fuse with the lysosomes, and next later all the food particles are going to be digested by the lysosomes. Yes, next one is pinocytosis. Okay, pinocytosis is also called as cell drinking process. Okay, I am writing here cell drinking. Okay, cell drinking process. So here the extracellular, in the extracellular fluid, whatever the solutes and solvent molecules are going to be present, if these solutes and solvents which are present outside the cell, if it wants to be transported from outer to inner, okay, so this process is called as a cell eating process, where it is going to take all the fluid, all the solutes and solvents from the outer cell environment to the inner cell environment. Okay, so now let us study about this pinocytosis, which is also called as cell drinking process. Okay, so here are some solute molecules. Okay, this is the step 1. In the step 2, again same plasma membrane is going to cover. Okay, plasma membrane is going to try to cover the solute molecules, and next it is going to form one vesicle, and it is going to store these solutes in that, and next in the step 4, this vacuole, whatever it is going to form, it is going to pinch off along with the solute molecules, and next later these are going to be freely distributed inside the cytoplasm. Clear with the concept pinocytosis and phagocytosis? In the pinocytosis, it is called as cell drinking process, because there some extracellular solutes and fluids, whatever those are going to be present, the plasma membrane is going to invaginate, it is going to form the vesicle, and then next the vesicle is going to be pinched off, and then next it is going to release all the solutes inside this inside this cytoplasm. Yes, this is about the endocytosis. Yes, next now we will see how the exocytosis process is going to occur. Yes, this is the exocytosis. Yes. See, in the endocytosis, when it comes to the phagocytosis, that is nothing but cell eating process, the amoeba, after capturing the food molecules, after fusing with the lysosomes, the lysosomes are going to digest the food particles. So sometimes there will be some undigested residues present inside the cell. Okay. So the cell, after, see, by seeing this diagram you are going to understand. See, it is a cytoplasm and it is a vesicle, okay, which is going to contain some undigested residues, okay, which is going to contain some waste cellular debris, and next here it is going to form, it is the first stage which is going to contain the cellular waste materials. In the next stage, it is going to create a vesicle called as secretory vesicle. Okay, it is going to form the secretory vesicle, and then next it will form the inward movement. Okay, So, in the first in the endocytosis we have observed that the plasma membrane is going to invaginate. So, here the plasma membrane will push like this in the downward position. Yes, it becomes in this manner. Okay, the plasma membrane will just, it is going to invaginate in the downwards position, and next whatever the waste materials which are not required for the cell, those are going to be secreted outwards freely from the plasma membrane. Okay, clear children? This is how the exocytosis process is going to occur. So, now here this exocytosis is also called as cell vomiting process. Okay, cell vomiting process. It is nothing but the undigested residues which are not required for the cell are going to be transported outside to the plasma membrane by creating the secretory vesicles. Okay, clear with all the concepts, clear with the concepts like plasma membrane, its composition, its mode of transport mechanisms. We have discussed in detail about the osmosis, diffusion, active mode of transport and passive transport. We have also discussed the extra information in the passive transport, that is nothing but simple diffusion and facilitated diffusion. When it comes to osmosis, we have studied in detail about the types of solutions with respect to the cell. Those are isotonic solution, hypertonic solution and hypotonic, hypertonic and isotonic type of solutions. And next we have also discussed in detail about exosmosis and endosmosis. Now just now whatever we have discussed, this is all about the bulk transport mechanism across the plasma membrane, which is going to contain the endocytosis and exocytosis. Clear with all the concepts, children? So, please stay tuned for the next live session. So, from the next live session, in the next class we are going to learn in detail about the cell organelles, children. Okay. So, the lesson, the chapter is going to form more interesting from the next session. This is all about the structures whatever we have studied. Next we are going to study about the all the cell organelles like nucleus, lysosomes, mitochondria, next chloroplast, okay, and the next Golgi body. These all structures, these all functions along with the structures, diagrams we are going to study in the next session. So, please stay tuned, like, share and subscribe to Neet World. Thank you.